Uh, Good afternoon. Thank you for inviting me. Sorry due to some circumstances out of my control, I wasn't able to make the meeting, but I've sent this video of my talk. Hopefully, be useful. So I'm going to talk about uncertainties and radiation predictions, focusing on galactic cosmic rays. Okay, so the acute risk I won't talk about because they're really an operational issue. We've done many studies showing that they can be shielded against. So as long as you have alert dosimetry and a good shielding approach, you're safe from solar particle events. There is a residual risk of cancer. So the main focus is late effects include cancer and several non-cancer risks, most importantly cognitive detriments, perhaps circulatory diseases and for longer missions, cataracts. For most missions that are shorter duration, say less than 90 days, even the cosmic rays are just a minor concern. You're not pushing against any health standards or limits. So the large uncertainties in the cancer and the cognitive risk and perhaps other ones is the main issue. And point out the timeline for these studies is long to iterate studies, lifespan studies in small animals, et cetera. So we mentioned that you should have limits because the acceptable risk should be weighed against the importance of a mission. So we've done studies using the 3% at the 95% confidence interval, and now I think that's open for debate whether that's the criteria or not. To me, it's a good criteria because the risk of flight failure is much less than that. And when you adjust for life loss from a cancer death compared to a mission loss, being 3 times higher seems like a reasonable estimate. I'll just give a little bit introduction about the design environments and shielding, and then because these uncertainties are much smaller, these things aren't known perfectly, but the uncertainty is standard error is less than 15%, which we've shown in comparison to space station, space shuttle, um, NASA Mir missions, and also the Mars Odyssey and Mars Marie missions. So time and time again, we can predict the quantities like dose and dose equivalent within 15% accuracy. There are some areas that could be improved, but these are much smaller than the radiobiology uncertainties. Okay, so the environment is well known. There's many balloon and satellite measurements. The Voyager 1 measurements now outside the heliopause give us a good understanding of the environment without solar modulation. So this chart just shows you our model for the cosmic ray spectrum at different time periods and also the LIS, local interstellar spectrum. And you can also see that the energies, this is a little bit misleading. It's on a log scale for the energy. The median energy of heavy ions is 1.5 GeV per nucleon. So I hear people talking about the Brookhaven NASA Space Lab doing simulations of cosmic rays, which is an odd idea in the first place because you're not exposing humans, you're exposing models, cell or small animal models that should be asking a research question. You can't measure risk. You have to use models to understand the risk. But the one point is then, so the NSRL can only get you half the GCR flux because half of it is above 1.5 GeV for the heavy ion. So it's a poor simulation. And then you get into the temporal aspects. It's not very close to temporal patterns of dose rate for a human exposure in a long mission. Okay, then the shielding, so solar particle events can be shielded, and we have a lot of information on what type of material gives you the best shielding, so hydrogen-rich materials. But even then, you have on this chart shows you have a initial decline at solar max if you have a large SP, and even some decline at solar min where it's just cosmic rays. But then it saturates very quickly, and you can actually have a rise in dose equivalent if you have too much shielding because of the secondary. So these curves are relatively flat except for the pure hydrogen, and this is because your gains and losses are in equilibrium. This just shows you that some predictions of the Bragg curves for different heavy ions, showing you how well we can reproduce them with the cross-section models I developed over the years. So very good accuracy, and it shows you secondaries are even at very small depth at these higher energies are contributing very substantially to the depth dose curves for these heavy ions. This just shows an example how well we did compared to the Mars Odyssey experiments with the rad detectors. So here, even less than 10% agreement. Okay, we developed a model, the quality factor that's still used at NASA. And what we did is we noticed that radiobiology experiments that have looked in detail show that quality is not a function of LET alone, but it also depends on the charge and the velocity. And this is also predicted by various theoretical models based on track structure, including detailed Monte Carlo or radial dose models. And so what we did was we formulated a functional form that's similar to what would be predicted by either the frequency distribution in a nanometer size volume or a radial dose model. And then we fit the parameters of the model to mouse tumor data, chromosome exchanges, and neoplastic transformation. So this is essentially all the cancer or surrogate cancer model data that in the world for heavy ions, we compared that and then fit the model and got good estimates of the parameters. And then the parameters have PDFs that we can sample across to get the probability distribution, uncertainty distributions. And one thing we noticed is that all the data that tested at less than 1 ion per cell with multiple doses shows that heavy ions have a deviation from a linear response at these low doses. So this is suggested of non-targeted effects. So I'll go ahead and call that targeted effects dose response model. And I won't go into all the past studies on mechanisms of non-targeted effects, but they would also suggest that you would have this deviation from linearity in a harmful way at very low doses. So now if you do an experiment with just one dose, one low dose, you won't really see it. You have to use multiple low doses. Okay, and there's another picture of the quality factor showing you that it has two terms, a penumbra term that's affected by dose rate, and then a core term that's not affected by dose rate. And that's important to note because the uncertainty in the dose rate modifier, I'll call it a DDRAF, is substantial. So having this these two relative terms reduces your uncertainty because obviously the core term where the doses are hundreds to thousands or even 10,000s of gray in a microscopic scale would not be affected by the dose rate. The penumbra term is affected because the experiments are using fluences where the tracks would overlap from different particles because of these long range delta rays. Okay, so the qualitative difference is the main issue, and non-target effects falls under that category. So you have these nonlinear things, but also the types of cancer, the shape of the dose response indicate it's not just a scaling factor like an RBE or quality factor. There's more to it than that. I won't go to all the experiments, but anything that has really tested, this suggests that more aggressive tumors and different types of tumors are occurring with heavy ions compared to gamma rays or even high energy protons. And this is an example of what I was talking about with the low dose response. So on these three upper curves, we have chromosome aberrations, chromatid aberrations, or a neoplastic transformation plotted versus dose. And then if you take these upper curves and divide the x-axis by the number of traversals per cell, and then scale it to the size of the cell that's the target, you get these lower curves. And you can see as soon as you get below 1 traversal per cell, the response shoots up. So if it was a linear response, it would just be a horizontal line. And we're showing that below 1 track per cell, you're getting this nonlinear increase because it's not a horizontal line. And this was also true for the Harderian gland experiment. So the Harderian gland is not the most logical one to represent humans, but it's the only one that used multiple low doses. So these are all data from Michael Fry and Ed Alpin, and also a more recent paper by Polly Chang tested this. So what you see is that as soon as you get below this 1 track per cell, the area of the target cell in the Harderian gland is a little bit less than 30 square microns. You get this shoot increase where it should be a horizontal line if it's a low dose, if it's a linear response. And this kind of makes sense because if you look at a detailed picture of the track, this is for high LET iron particles. This is the vertical and the horizontal view. And you think about cell death. Now, if you average over all the impact parameters, the cross section is not greater than or even approaching the unity scale to the area of the cell. Usually, for heavy ions, it's about 20 to 50% probability of a cell death for a single particle. But when you think about the impact parameter, you can see that that cross-section average over all impact parameters doesn't tell you what happens when the particle goes through the cell. So we would think that it has very small likelihood that if the particle goes through the cell nucleus that it survives. It would be a much less than 1% probability. If you see these very high tumor responses, it must be from either the delta rays or the non-targeted effects. So we looked at the delta ray part, and compared to the gamma ray response for this system, it wouldn't come close to producing the number of tumors when we match the delta ray dose to the gamma ray dose. So we think that's another indication that something else is going on that we'll label as non-targeted effects. So the non-targeted effects then busts your whole scheme of whether you're safe or not, because you're now looking at a very high RBE. So if you're going to use an RBE model, instead of using 10 to 20 for heavy ions, you might be approaching 100. So this is some prediction showing you comparing with or without the non-target effects. Here, what I did, I took the HC trend code with my cross-section models and my GCR environment and made some predictions with or without non-targeted effects. And you can see it's a little bit more than double, and also pushes the uncertainty well above 10%. So that's very problematic. So now you're talking about uncertainties that would be shunned upon to say it's safe for a group. Now, the RBE approach and quality factors is based on scaling to the gamma ray data from the atomic bomb data. And there's a few other gamma ray or X-ray cancer data sets. And to me, this is also a problem. So I thought about, well, what if the atomic bombs had never been used and we had no data? And actually, that's what would happen in most of medicine. So in medicine and toxicology, you usually use some type of relative risk model to use experimental data to predict risk. So I put together a model, which I call DERR, Direct Excess Relative Risk Model, and then went through all the animal data for heavy ions and fission neutrons and fit a response there, and then use that directly with the background cancer rates for different populations to make some predictions. So one of the things, as I said before, most of these heavy ion and fission neutrons show some type of bending in the response at lower doses. So I compared a linear model to this, what I call saturation model. So the linear model is this alpha parameter and the saturation, this A and B parameters, and then fit the various data sets. And here's some examples showing you. Overall, the saturation model agrees much more with the data than this linear model. So in a lot of cases, the linear model doesn't fit the data, and in just about all the cases, one exception would be liver cancer and one study with fission neutrons, but in the other studies, it does fit the data very well. And so they made some predictions, again, with the HC trend code for with my GCR model and cross sections to look at predictions. And what we found, so here we can only make predictions for tissue-specific cancer rates. So for lung, breast, and liver, we had available animal data. So we avoid RBEs and the atomic bomb data altogether and just scale relative risk to the background cancer rates for the populations under study. And you can see this first column is without the non-target effects. This is with it, and then this is this direct application of ERRs. And you can see in most cases several of the cases with non-target effects agrees with the DER. The exception would be the lung cancer, where we're getting a much lower prediction with the DER model. And so I found this to be interesting because this is controversial, this difference between males and females in the atomic bomb survivors, and also some of the Russian epidemiology studies with radiation, that there seems to be a higher risk for females but mechanistically, there's no good explanation for that. The one thing that comes up is the interaction of radiation and the use of tobacco products, especially for people that smoke that low to moderate amounts of cigarettes, that there seems to be an interaction from the results from the Hiroshima and Nagasaki study. So with animal models, you avoid this problem altogether because there's no influence of smoking at all in these studies. So you can see, so this is, to me, this is interesting that you're predicting a much lower risk with the direct application. And then for the other tumors, breast and liver, the direct application of relative risk agrees with the non-targeted effects model, and both of those are much higher than the standard model using the quality factor and the atomic bomb survivor data for the gamma rays. Now I'll just talk a couple minutes about the non-cancer risk. So there's quite a few of these come up, but first I want to point out is if you maintain a cancer risk limit, you're really protected against most of these things that would occur. Yeah, you're limiting your risk of cancer, and most of the studies for circulatory disease risk, we've also published that say that risk would be about a third of the cancer risk. So you have a somewhat of addition, but the harm or lethality is less because it comes later in life. So the only one that stands out is that might not protect you is the cognitive risk. All these other things would be essentially protected by having a strong cancer risk limit. Now, the CNS radiation injury, this is something that we were kind of felt good about at NASA because we were really early on and with Walter Schimling and myself funding studies of this. Early on, it was Bernie Rabin and Jim Josephs and then John Fike and Charlie Lamoli and Kerry O'Banion and others, Richard Britton, a few other people. And they really opened the door of like, is this a problem? It's different than what you would see in radiation oncology because the effects are not from apoptosis or necrosis. It has to be something else. So inflammation, perhaps. Charlie Lamoli and also John Fike before that talked about changes to dendrite properties, losses of spines and dendritic length. But these things are really hard to prove at low doses. Impaired neurogenesis, we looked at all that data and said, well, it's true at 1 gray, but it's not really important at all at 0.1 gray. I remind you, the heavy ion dose on a Mars mission is less than 0.1 gray. So it has to be something else. So inflammation is still possible, but and perhaps the spine loss, but there really isn't a strong indication of what a mechanism would be. So we did some meta-analysis of all the novel object recognition data, found that you could fit a nice response on radiation quality, even if the data looks kind of noisy. And you see that this is relative risk. So what we did is take the risk or the background and then develop a relative risk as a function of radiation quality and dose. And then this will show you some of the data. So now one of the things that you point out is that it's really investigator dependent, that people are getting different results with different models and different types of radiation. And some of them have very nice dose response. For example, even for proton, you have less data, but it seems like a linear response here for silicon. Other ones, it's very noisy with iron. So maybe one investigator sees a nice response, but other ones are seeing kind of a noisy response. But we fit a model. You can read our paper, was in Rad Research, and then we followed that up with some predictions for space missions. And what we found is that if you do this meta-analysis, you can make a prediction of the relative risk for novel object recognition increase during different types of missions, but it's very small. So, and we compared that to this is the most common cognitive test used in animal models, and people have done this in Alzheimer's disease models, Parkinson's disease models, and also traumatic brain injury models. And you can see, so they're seeing quite a large increase in risk. We're predicting a very small increase in risk. Now, there's other endpoints that other types of cognitive tests that people can pursue, more sophisticated one, but this is the standard one. This is some data for conditional fear conditioning. Again, a lot of negative results that Jacob Raver has reported. And you can see there's no clear indication of an elevated risk. Now, if you go to other endpoints, you'll see an elevated risk, but they're kind of model specific. So I think that's one of the open questions is the models that are being used. They're really in the mouse, it's 90% of the data was C57 black. And in rats, it's usually Wistar or Sprague-Dawley. So there's been really no genetic studies. I think Jacob Reber has one or two studies, but it's not one really conclusive on how you would approach the genetic background and how that impacts cognitive tests and whether these well-known strains that have been used are they the most sensitive or the most resistant strain? We don't know that, and also the underreporting by people since it's difficult to publish negative results, and also the variability across investigators. So in conclusion, we would say that short duration missions, the GCR seems like it's a manageable risk, say less than 90 days. SPs could be or should be well protected if you have a proper dosimetry and shielding, and the shielding that you need is manageable. But for these cancer risk, you could go above 10%, maybe uncertainties as high as 30% for morbidity. And shielding doesn't really help you from the GCR. Reducing biological uncertainties might reduce the risk estimate, so that's good. But it also would be the same information that would lead to mitigation, say biological mitigation approaches. Additional CNS effects will not be understood without extensive animal studies and studies of mechanisms. It's really old hat now to do cognitive testing with a different beam or this so-called GCR simulator, a different cognitive test, a different dosage. You really need to tie the mechanism in there because that's the only approach to extrapolate to humans. So the approach to protect against the upper 95% confidence level, I still think that's strong. I know NASA has dropped that. They essentially dropped their limits. They said they'll use a 0.6 sievert limit for everything, and then independent of male and female age. And then if they approach the 0.6 sievert limit, maybe they'll just drop that too. So it's sort of like, in my mind, it's sort of like not having a limit. Lack of knowledge on CNS risk is important. This circulatory disease risk, I think it's there, but it's only going to be a fraction of what the cancer risks are. So the cancer risk limit seems to be the key for most of these things, with some open questions about cognition. So I'll end there, and thank you. I hope the meeting is a success and people enjoy their visits to India.